Welcome to this tutorial video. So in this video we're going to answer some tutorial sheet questions on unit 3, specifically unit 3.1, that is talking about fatty acids metabolism. So we'll start right away. So the first question in this tutorial sheet is as follows. Fatty acids are carboxylic acids with hydrocarbon side chain. They are the simplest form of lipids. Fatty acids are carboxylic acids with hydrocarbon side chain, and they are the simplest form of lipids. Question A. So question A is as follows. Distinguish between ketogenesis and lipolysis. Distinguish between ketogenesis and lipolysis. So I'll just define the two, then I'm going to make a distinguishing statement at the end. Yeah. So let's start with the ketogenesis. So ketogenesis, this is a biological process that takes place in the liver and involves the production of ketone bodies or ketones. Ketogenesis, this is a biological process that takes place in the liver and involves the production of ketone bodies or ketones. That is the definition of ketogenesis. Now let's define lipolysis. Lipolysis, this is a biological process that involves the breakdown of what? Triglycerides in order to produce free fatty acids and a glycerol molecule. Some of you may be wondering to say, why am I saying triglycerides? Why didn't I say lipids in general? So usually in biochemistry, when we're talking about lipolysis, of course, you can generally say it's a breakdown of lipids, but we specify to triglycerides in biochemistry mostly. So we're saying lipolysis is the breakdown of what? Triglycerides in order to produce free fatty acids and glycerol. So our conclusive statement would be as follows. Ketogenesis is the biological process that takes place in the liver and involves the production or formation of ketone bodies or ketones, while lipolysis is a biological process, of course, that takes place in the adipose tissue and involves the breakdown of what? Triglycerides in order to produce free fatty acid and a glycerol molecule. So that is the first question that we were answering, the difference between ketogenesis and lipolysis. Let's go to the second question. Question A, of course, on question 2 there. So question 2 is as follows. Differentiate or distinguish between ketosis and ketoacidosis. So before I even start differentiating the two, actually, the definitions of if you want to define ketosis and ketoacidosis, they are almost the same, 99% the same. The difference is when these processes are taking place and in what type of people. You understand? So, when we're talking about ketosis, I would start with ketosis. So, we are from defining ketogenesis, okay, which we said is simply the biological process that involves the formation of ketones or ketone bodies. Now, you must understand that those ketones or ketone bodies, they are acidic. They are acidic. So, meaning if they are acidic, if they are produced too much in the body, they are going to decrease the pH levels in the blood. Because remember, the more acidic the substance is, the more the pH is going to drop. So, if you're talking about ketosis, we're just talking about the elevated or the increase of ketone bodies in the blood that causes the drop in pH. And you must understand that that is very, very bad, because if the blood becomes acidic, okay, it can start damaging organs, okay, it can damage your brain, it can damage your kidney, it can damage your liver and different organs. Now, when I'm talking about ketosis, so what I was explaining is the same both in ketosis and ketoacidosis. There is increase of these ketone bodies in the blood that causes the blood to be acidic and the pH is going to drop. Now, in ketosis, we are going to generalize it such that we are going to say this one, the ketosis takes place in people or patients who are just starving. Okay, since they are starving, there will be breakdown of what? Of fats. There will be breakdown of triglycerides. Those triglycerides are going to produce free fatty acids. Those free fatty acids are going to start undergoing beta oxidation. Now we said that in the liver, when there's beta oxidation in the liver, when there's too much beta oxidation in the liver, because this person is starving and they're depending on fats, that excess acetyl-CoA that is being produced in beta oxidation, remember beta oxidation is just a process that involves the breakdown of fats in order to produce acetyl-CoA. So that excess acetyl-CoA, it will now start forming the ketones, and that's what we refer to as ketogenesis. So if you want now to come up with the overall difference between the two, if you're talking about ketoacidosis, okay, it is the same process. It is the same process that involves, the increase or the elevation of ketone bodies in the blood that makes the blood to be acidic. Now, ketoacidosis, we give this term, or we use this term when we are talking about diabetic patients. Remember, diabetic patients have got decreased insulin levels, that is in diabetes type 1. Then in diabetes type 2, usually you find that the cells don't respond to insulin. So we're going to stick to type 1 in this particular video, because in type 1 we know very well to say there is no insulin. The pancreas is not secreting insulin. So if the pancreas is not secreting insulin, it simply means what? Their blood glucose levels are going to be very high, because there is no insulin to be able to regulate those blood glucose levels. So what does it mean in this diabetic patient if they don't have insulin? If they don't have insulin, it simply means that their blood glucose levels are going to be high. Too much glucose in the blood is going to cause too much glycolysis. Too much glycolysis is going to producing a lot of pyruvate. That pyruvate will be producing a lot of what? Acetyl-CoA. And remember, when you've got acetyl-CoA in excess, it will start producing ketones in ketogenesis. That acetyl-CoA will start producing what? Ketones. So those ketones, again, like I said, they're going to make the blood to be what? Acidic, because ketones are acidic in general, and they're going to drop the pH. So the difference is that everything is everything is the same in terms of definition, but ketosis, we just generalize it to people who are starving, people who are not in a good well-fed state. Then ketoacidosis will just specify to people who are diabetic. So now let's come up with the general, conclusive difference. So ketoacidosis, this is a situation in the body that involves the increase of ketone bodies in the blood, which makes the blood to be acidic in people or patients who are starving. While ketoacidosis, this is a situation that is characterized by the increase of ketone bodies in the blood, which causes the blood to be acidic in people or patients who are diabetic, specifically diabetes type 1. I'm going to repeat. Ketosis, this is a situation that is characterized by the increase of what? Ketone bodies that causes the blood to be acidic, and this takes place in patients or people who are just starving or who are not in a good fed state. While ketoacidosis, this is a situation that is characterized by the increase of ketone bodies that causes the blood to be acidic, okay, and it is taking place in people who are diabetic, specifically diabetes type 1. That is the answer for the second question. Now, talking about the third question, differentiate between ketone bodies and autoantibodies. So this question we are just trying to, just more like tease you to just see if you understand biochemistry very, very well. So ketone bodies and autoantibodies, they are two different things. Like I said, ketone bodies, these are energy source molecules that are produced from excess acetyl-CoA. I've already described how these ketones are formed. When you've got excess acetyl-CoA, you start producing ketone bodies. And remember, in as much as these ketone bodies are acidic or not good for somebody's healthy, but at some point they are used as energy sources by the brain, by the heart, or skeletal muscles. Okay, they are used. Now, when talking about autoantibodies, these are just antibodies that attacks one's immune system. Imagine you've got your immune system that protects you against infection. Then you find that those antibodies are the ones that protect you against infection. Now you find that these antibodies, they start fighting against you. No wonder we're saying auto, because they are fighting against the very person. So in autoantibodies, you find that the body will start producing antibodies that start damaging one's own tissue. I can give an example in diabetes. Okay, you find that when someone is diabetic, there is this auto antibodies response. What happens is that these antibodies, they'll start attacking the pancreas cells that produce insulin, and this person won't be able to produce insulin. So that's what happens in what? In autoantibodies. It's just basically when somebody's own immune system is attacking the very person, is attacking oneself. So what is the difference? Ketone bodies, you just say ketone bodies, these are bodies or rather molecules that are produced from excess acetyl-CoA, and they can be used as energy source molecules during starvation. While autoantibodies, these are antibodies that fight against one's immune system, okay, or that fight one's own body system. That is the difference. So we're talking about ketone bodies, we're just talking about energy source molecules that are produced from excess acetyl-CoA and can be used as energy source molecules during starvation, or when starvation is too much. Then autoantibodies, these are antibodies that fight against one's, or rather against one's immune system. That is the difference between the two. Now, question B. Question B there, the question is discuss the formation and metabolic functions of ketone bodies. Discuss the formation and metabolic functions of what? Ketone bodies. So you must understand, how are these ketone bodies formed, and what is the metabolic function of these ketone bodies. So let's start with the first part. Discuss the formation of what? Ketone bodies. So how are the ketone bodies formed? So this is how you're going to describe the process. So ketone bodies, first to define ketone bodies, these are energy source molecules that are produced from excess acetyl-CoA and they form ketones. Okay. Now, how are they formed? So you start explaining like this. When somebody is in a low-fed state, when somebody's glucose levels are low, okay, the body will start breaking down fats, okay, in form of it will start breaking down triglycerides. When the triglycerides are broken down, they are going to produce free fat acids and a glycerol molecule. The free fat acids that are produced from triglycerides, they are going to be transported to the liver, to the muscles, and they will start undergoing beta oxidation. So those free fat acids, as they are undergoing beta oxidation, they'll be producing acetyl-CoA. Now, if this person keeps on, being in a low fed state, or if their blood glucose levels keep on dropping, the body will keep on breaking down those fats. And if they're breaking down those fats, the body will start producing a lot of acetyl-CoA. Now, that excess acetyl-CoA, specifically in the liver, that excess acetyl-CoA in the liver will start forming what we call ketones. So how are these ketones formed? The first step is that from that excess acetyl-CoA, the first step is that you're going to combine two acetyl-CoA molecules. Okay? You're going to combine two molecules of acetyl-CoA using the enzyme that we call thiolase. And then you're going to form what we call acetyl-acetyl-CoA. The first step in the formation of ketone bodies, you're going to combine two molecules of what? Acetyl-CoA, and then you're going to use the enzyme that we call thiolase. Then you're going to form what we call acetyl-acetyl-CoA. You know that in that step we've lost one CoA because we had acetyl-CoA from another molecule, acetyl-CoA from another molecule, but when we combine, we're just forming acetyl-acetyl-CoA, meaning we lost one CoA. And then the enzyme that we used in combining those two molecules of acetyl-CoA, we used what we call thiolase enzyme. After that, you are going to get another extra acetyl-CoA. You combine it with the just formed acetyl-acetyl-CoA. Now, what enzyme do you use? Use what we call HMG-CoA synthase. Remember, after you formed your acetyl-acetyl-CoA in the first step, and then you're going to get an extra CoA and combine it with that acetyl-acetyl-CoA. What you want to form is what we call HMG-CoA. No wonder the enzyme was saying it's HMG-CoA synthase. What does HMG mean? It means 3-hydroxy-3-methyl-glutaryl-CoA. That is the HMG. That's where it's coming from. So, what have we said? In the first step, you combine two molecules of acetyl-CoA using thiolase and you form what we call acetyl-acetyl-CoA. The second step, you get the acetyl-acetyl-CoA and you add another CoA using what we call HMG-CoA synthase. And in that step again, you're going to lose another CoA because HMG only has one CoA. But you got your acetyl-acetyl-CoA, which is the CoA, then you added another acetyl-CoA. So meaning you're supposed to have two CoAs, but you're going to lose one. That's why you're just going to form HMG-CoA, 3-hydroxy 3-methyl-glutaryl-CoA. That is in the that is in the second step. Now, in the third step, after you form your HMG-CoA, okay, using of course HMG synthase enzyme. After you form your HMG-CoA, what you're going to do now, you're just going to remove one acetyl-CoA from that HMG. Using the enzyme that we call HMG-CoA lyase enzyme. HMG-CoA lyase enzyme. So just going to remove one acetyl-CoA from that HMG. Then you're just going to remain with what? Acetoacetate. That acetoacetate is a ketone body already. That acetoacetate that you formed is already a ketone body or a ketone. So that's how these ketones are produced. In summary, we've said when you're forming ketones, these are produced when, actually we're supposed to even specify, these are produced in the liver as the primary organ of ketogenesis. When there's too much beta oxidation that is producing too much excess or too much acetyl-CoA, that excess acetyl-CoA will start forming ketone bodies. The first step we said we are going to combine 2 molecules of acetyl-CoA using thiolase enzyme, then we form what we call acetoacetyl-CoA. After we form acetoacetyl-CoA, we get another CoA, we add it on the acetoacetyl-CoA, we combine them. Then we form what we call HMG-CoA, using what we call HMG synthase enzyme. After you form your HMG-CoA, you're going to remove a CoA. You're going to remove acetyl-CoA from that HMG. Then you just remain with what? Acetoacetate, which is just one of the ketone bodies. That acetoacetate now can be used to form the other two types of ketone bodies, just using spontaneous reactions. For example, acetoacetate can form acetone, which is the second type of ketone bodies. Then acetoacetate can also form what we call beta-hydroxybutyrate, which is just also another ketone body. So the main ketone bodies that we're going to form are acetoacetate, acetone, and beta-hydroxybutyrate. Now, we've described how these ketone bodies are formed. Now, let's talk about the metabolic functions of these ketone bodies. The functions of these ketone bodies, I already told you, they are used as energy source molecules during starvation. So when somebody is starving too much, because remember, when they're starving too much, the body will keep on breaking down those fats. It will keep on breaking them down. And then they'll be producing a lot of acetyl-CoA. So imagine in a situation whereby maybe all the fats were almost depleted. These ketone bodies now, they are the ones that the body is going to reverse to. It will start now saying to say, I think I was producing some molecules when I had too much acetyl-CoA. Let me go back to them. So the body is going to start using these ketones now as energy source molecules. Your brain can use these ketone bodies, okay? Your skeletal muscles can use these ketone bodies, all right? As well as your cardiac muscles can use these ketone bodies if the starvation is too much. So how are they using them? They're going to start converting those ketone bodies back to acetyl-CoA, and that acetyl-CoA now can be used in the Krebs cycle, produce NADH, produce FADH2, which can now be used in the electron transport chain and provide what? ATP. So this is how these ketone bodies are used. They are used as energy source molecules. If the starvation is too much, they'll be converted back to acetyl-CoA again. Then acetyl-CoA used in the Krebs cycle, produce NADH, FADH2 that are used in electron transport chain and provide ATP for the skeletal muscles, cardiac muscles. So that is the end of question B. All right. Having looked at that, let's go and answer the questions on the next slide. Now, the next slide, which is just talking about question C, describe the role of insulin in lipid metabolism. Describe the law of insulin in lipid metabolism. So we're talking about lipid metabolism in general. In biochemistry, we're just talking about the breakdown of what? Triglycerides. So describe the law of insulin in lipid metabolism. So, do you think insulin is going to promote lipid metabolism? Of course, when you are talking about, if you wanted to specify in terms of lipids, okay, you can talk about it. You can talk about fatty acids and talk about how insulin does that process, whether it promotes or inhibits it. If you want, you can talk about triglycerides, whether insulin promotes or inhibits that process. But since we're talking about fatty acids, and we know that fatty acids as well as lipids, they all fall under lipids. So we're going to mention everything. Okay, so that you are just able to get all the full marks. So let's start with first the fatty acids. Okay, let's start with the fat acids. So, since fat acids, they fall under lipids, of course. So these fat acids, do you think, because when we say metabolism, we're talking about their breakdown and building. Okay, so I'll start with the fat acid for fat acid synthesis. So I know very well to say, if I've got too much blood glucose levels, if somebody has got too much blood glucose levels, meaning there will be too much insulin. So if too much insulin is there, there will be too much glycolysis. Too much glycolysis, too much pyruvate. Too much pyruvate, too much acetyl-CoA. That excess acetyl-CoA, we said, if somebody is in a good fed state, that acetyl-CoA can be used to form fatty acids in fat acid synthesis. And remember, when you're talking about the most degraded step in fat acid synthesis, which is the conversion of acetyl-CoA to malonyl-CoA, that is catalyzed by the enzyme that we call acetyl-CoA carboxylase. That step is promoted by insulin. So insulin is going to promote the formation of what? Fatty acids by promoting the activity of what? Acetyl-CoA carboxylase. Basically, what it does is that it dysphosphorates it. It removes phosphate groups on that acetyl-CoA carboxylase and makes it active so that it can convert acetyl-CoA to malonyl-CoA. So basically, in fat acid synthesis, insulin is going to promote that process. And in the opposite, of course, beta oxidation, it will not promote, insulin cannot promote beta oxidation, because imagine, somebody has got low blood glucose levels. Why do you think insulin is going to promote the breakdown of fats? No, because insulin likes building things. It doesn't like breaking things. So in beta oxidation, insulin cannot promote that process. Now let's talk about the role of insulin in triglycerides catabolism, in the breakdown of triglycerides. Do you think insulin is going to promote this process? Of course no, because you are breaking down things. Generally, insulin doesn't like, breaking down stuff. It's like building things. Okay, so insulin will not promote the the breakdown of what? Triglycerides, because what promotes the breakdown of triglycerides is what we call glucagon. And glucagon and insulin are just more like cousins. They do the opposite of what the other one does. So in general, when we're talking about lipid metabolism, insulin is just going to promote the formation of what? Where it's just going to promote the formation of what? Fatty acids. The fat acid synthesis is going to promote that process. If you want, you can even talk about insulin promoting the formation of what? Of cholesterol, or cholesterol synthesis. Because when you're forming cholesterol, it simply means you've got excess acetyl-CoA, because there's no way you can be using acetyl-CoA instead of making it go into the Krebs cycle, start forming other things if you've got too much energy. If you've got low energy. So if you've got too much acetyl-CoA, you can use that acetyl-CoA to form cholesterol, or you can use it to form what? Fat acids. And insulin is going to promote those particular pathways. The only thing that insulin is going to inhibit is beta oxidation. It's going to inhibit beta oxidation. It's going to also, inhibit triglycerides catabolism. So that is on the first part. Going on the second question, describe the role of lipases in the degradation of what? Triglycerides. So triglycerides, these are just the triglycerides. They're just triglycerides in general. So what is the role of triglyceride? The lipases enzyme in triglycerides. So I already told you to say, when I'm talking about lipases enzyme, you're talking about those enzymes that break down triglycerides. So you just go straight. So the lipases enzyme, remember I gave you three types. You've got adipose tissue triglyceride lipase enzyme, you've got hormone sensitive lipase enzyme, and the monoglyceride lipase enzyme. Their basic role is to break down triglycerides in order to produce glycerol and free fat acids when somebody has got low blood glucose levels. That is the primary function of these lipases enzyme. If you want, you can just generalize them to say lipase enzyme. But if you want to go into the specific types, you can say under the lipase enzyme, I've got adipose tissue triglyceride lipase enzyme as one type. I've got hormone sensitive lipase enzyme. I also have monoglyceride lipase enzyme, which are just used in the breakdown of what? Triglycerides, in order to produce free fat acids and glycerol. So their just general function is to be able to break down these triglycerides. So we're done with that particular question. Now, describe the role of what? Acetyl-CoA in fat acid synthesis. This is a very simple question. So acetyl-CoA, we said when you're producing fat acids, we can only do that if we've got what excess acetyl-CoA. So acetyl-CoA is more like the substrate that you need to be able to form these fat acids. So what happens is that, for example, when somebody is in a well-fed state, they've got too much glucose, too much pyruvate in glycolysis, too much pyruvate, too much acetyl-CoA. Too much acetyl-CoA now, that acetyl-CoA in the Krebs cycle, it will combine with oxaloacetate and form citrate. We talked about how citrate will be able to be shattered or removed from the what? From the mitochondria into the cytoplasm. And then in the cytoplasm, citrate is going to be converted back to citrate and oxaloacetate using the enzyme that we call ATP citrate lyase enzyme. We talked about that. Now, when you have that acetyl-CoA now in the cytoplasm, it's going to be used to form what we call malonyl-CoA. So acetyl-CoA is going to be the raw substrate which we are going to convert from acetyl-CoA to malonyl-CoA using what we call acetyl-CoA carboxylase enzyme. When we do that, we form our malonyl-CoA. That malonyl-CoA now can be used to be able to form this palmitic acid or palmitate acid, which is just the fat acid. So the general answer should be acetyl-CoA is the raw substrate, okay, that the body uses to form fatty acids. It is used to form malonyl-CoA that can form palmitic acid or palmitate. That is the answer there. Then D, what is D saying? Give reasons to explain the need for the regulation of fat acid synthesis. So when talking about regulation of fat acid synthesis, so give reasons to explain the need for regulation of fat acid synthesis. Just think about it. Why do you think the body must regulate fat acid synthesis? What do you think? It's because if you've got too much fats, you understand, if you've got too much fats, then again that is going to cause a problem. So the body must regulate, it must create a balance between those particular fats that are being formed. Because if you've got too much fats again, that will not be very, very good for your health. Okay? So the body must regulate that. So, for example, you can talk about give reasons to explain the need for regulation of fat acid synthesis. The general reason is that the body must keep the fats in equilibrium, not too much of it, not too low of it. So it must balance that equation. How? By regulating that step that we talked about, the conversion of acetyl-CoA to malonyl-CoA, which is regulated by insulin, glucagon, okay, as well as citrate, ATP. Long chain fat acids, they regulate that process. The overall goal is to create a balance between the formation and the degradation of what? Fat acids, so that you don't have too much fat acids and you also don't have too low, fat acids. Because if you don't have, if you've got, let's say, less, if you've got less, fat acids again, that is going to be a problem, because these fat acids, they are part of, they are part of, for example, the phospholipid that are able to create, or that are part of the cell membrane. Okay, they are part of triglycerides. Okay, they are also part of different, components of lipids. So the whole goal is just to make sure that there is a balance between the formation of fat acids as well as the degradation of fat acids, so that it keeps everything in what? Equilibrium. Now, question E. Explain the stages and substrates involved in beta oxidation of fat acid synthesis. So I'll not go into details, but I'm just going to give you the overall, picture. So explain the stages of stages and substrates involved in beta oxidation of fat acids. So the substrate that you need, actually let's define beta oxidation. Beta oxidation, this is a biological process that involves the breakdown of palmitic acid or palmitate to produce acetyl-CoA. So what is the raw substrate that we need? Is what? Palmitic acid. So what are the steps involved? The first step is activation of fat acids. So we're going to activate fat acids. What are we going to use? Fat acetyl-CoA synthetase enzyme. When we're activating this fat acid, we're going to need fat acetyl-CoA synthetase enzyme. We're going to need ATP. We're going to need the CoA. So the first step is the activation of fat acid to form fat acetyl-CoA, or to form what we call acetyl-CoA. After the acetyl-CoA is activated, it will be transported from the cytoplasm inside the mitochondria. So what does that process involve? It involves CAT1 and CAT2. So I already explained to you what CAT1 and CAT2 simply means. Carnitine acyltransferase type 1, carnitine acyltransferase type 2. So after you activate your fat acid, it becomes a fat acetyl-CoA. CAT1 is going to remove the what? The CoA on the acyl-CoA and adds the carnitine, because as acyl-CoA cannot penetrate the inner membrane of the mitochondria. I think you know that. So Cat1 is going to remove the CoA and add the carnitine. Then with the help of translocase, that acyl-carnitine that has been formed to be able to penetrate through the inner membrane of the mitochondria. Then inside the mitochondria, since you have got acyl-carnitine, then Cat2 is going to remove that carnitine because you don't need that carnitine. Cat2 is going to remove the carnitine and add the CoA back. Then that carnitine will be transported back into the intermembrane space. Then you form your acyl, the CoA that you need for beta oxidation. After you form that acyl-CoA inside the mitochondria, okay, it will now be ready to undergo beta oxidation. I will not explain the what steps, okay, but just know that it will start undergoing now oxidation, hydration, oxidation, and cleavage, okay, and the cycle is going to repeat itself from oxidation, hydration, oxidation, and cleavage. It will repeat itself, okay. The total rounds, we say the total rounds of beta oxidation are at 7 rounds, which will produce 8 molecules of what? Acetyl-CoA. We looked at that. You can just add more details just to make sure that you get 4 marks on that. Now, talking about question F, what are the notable differences between fat acid synthesis and beta oxidation? Name the 2 potential consequences of ineffective regulation of the opposing process of beta oxidation and fat acid synthesis. So, let's just start with the first part. What are the notable differences between fat acid synthesis and beta oxidation? There are a lot of differences. So I can just give you these ones. So the difference is that in fat acid synthesis, there's too much glucose. Somebody is in a good fed state. In beta oxidation, there's low glucose. Somebody is in a low fed state. Okay. The other difference is that in beta oxidation, there is oxidation. And then in fat acid synthesis, there's what? There is reduction, okay? Then apart from that, in beta oxidation, there is hydration. In fat acid synthesis, there's what? Dehydration. You can give those differences. You can also talk about where they take place. Fat acid synthesis takes place in the cytoplasm. Beta oxidation takes place where? In the mitochondria. You can talk about those differences. Then the other part is saying name 2 potential consequences of ineffective regulation of the opposing processes. So if at all beta oxidation and fat acid synthesis is not being regulated, what do you think are the consequences? For example, if we're talking about fat acid synthesis, so I already told you about this part on I think on question D. So fat acid synthesis must be regulated. It's because imagine if you are just forming fats, they're not being regulated. Of course, you end up depleting the entire acetyl-CoA that is needed for the Krebs cycle, and then somebody is going to will not have enough energy because you've converted everything from acetyl-CoA to fats. No wonder there must be a balance. So if you deplete all the acetyl-CoA just in the need, all right, of just wanting to form fats, you're going to starve the body from energy, which is going to be bad because, for example, the brain needs ATP to be able to perform those cognitive functions. The heart will need ATP and all those stuffs. So there must be a balance between the formation and the degradation of fats, because if you are to convert everything into fats, because maybe there is no better balance of those insulin and glucagon hormones, then that is going to cause a problem. Okay, for example, there will be maybe you can talk about loss of cognitive function in the brain, irregular heartbeats in the heart. Okay, then beta oxidation, if it's not being regulated again, imagine you just keep on breaking down fats anyhow. You're just breaking down fats. If you're breaking fats down like too much, you're going to produce a lot of acetyl-CoA. Okay, and that is going to produce a lot of ketones. I've already explained that, which is going to cause ketoacidosis or ketosis. That is just increased amount of ketones in the blood, and the blood is going to be acidic, and then it will start damaging someone's brain, damage the kidney, damage the heart muscles, damage a lot of different organs. No wonder these hormones, the insulin, glucagon, epinephrine, they must come into play and regulate those particular pathways, because if everything is too much, then there will be problems and consequences. So that is what this question is trying to talk about. And that marks the end of this tutorial video, as we are just looking at the questions in your tutorial sheet.